Hello and welcome to Sick Notes. My name's Ed Hope. I'm a junior doctor in the UK, and on this channel we talk about medical things in simple terms. Today we're going to be revisiting one of our favourite shows, Cells at Work. This is episode number 9. Cool, so we've met the killer T cells before, or called the cytotoxic T cells. As a recap, cyto means cells and toxic means kill, so they're responsible for killing your own cells when they become infected with a virus or cancerous and things like that. The text here also mentions graft cells. So a graft is where in a medical procedure we take cells from somewhere and transplant them. Most people will automatically be thinking of transplant from one person to another. For example, an organ transplant, like a kidney transplant. That's the type of graft. We call this type of graft an allograft or homograft, taking tissue from the same species but not the same person. But you can also have grafts from your own body. For example, you may have a skin cancer and have the cancer surgically resected and have some skin taken from another part of the body to cover the area that was removed. This type of graft we call an autograft. What did the resected skin cell say to the other skin cells? Can I have your autograft? That is terrible. And finally, we have xenografts or heterografts, where we have a transplant from a completely different species. You may think, why the hell would we take something from a different species? Well, one thing we talked about in the last episode was problems with heart valves. And so tissue valves from pigs is a common heterograft. Grafting tissue from one place to another is a complex thing, mainly due to general surgical complications, plumbing in a blood supply, but the big thing is how the immune system handles the newly introduced graft cells. So we first need to closely match the donor and the recipient, and also put the recipient onto medications that dampen down the immune system. Medications that would be working on things like the cytotoxic T cells to stop it attacking the graft. Also worth noting that clearly in an autograft, so a graft from your own cells, the immune system isn't going to be a problem because it knows how to recognise your own cells. You miserable losers. Reminds me of the type of motivational skills I was subjected to as a medical student. I'm just kidding. The helper T cells, Baxter says here, when notified of invading external enemies, they serve as commanders, which is exactly right. They basically coordinate the type of response that you want the immune system to do, depending on the pathogen, the disease-causing thing that is coming at the body. So depending on the type of enemy they find, they'd secrete different chemical messengers called cytokines, which would activate different immune cells. For example, the immune reaction to a virus would require more of a cytotoxic T cell response rather than infection from a parasite, which would require more of a response from cells like the eosinophil. You may recall that all the cells in the blood are created in the bone marrow, a factory inside the center of our bones. So let's show how the T helper cell and the cytotoxic T cell fit into this family. So all the cells in the blood come from this stem cell, and we've already looked at how the macrophage, the red blood cell, the megakaryocyte that produce the platelets, the neutrophil, and the dendritic cell all come from this common myeloid progenitor cell. But there's another path that we haven't looked at that I didn't leave enough room for on the diagram. I didn't think we'd cover quite so many cells in this show. So instead of a myeloid progenitor cell, we get production of a lymphoid progenitor cell. This family of lymphoid cells include the natural killer cell that we've met before, the B cell that we've also met before. So the B cell is the one that produces antibodies. And finally, what we're going to learn about today, the T cells. So both the helper T cell and the cytotoxic T cell, although they look very different and have very different functions, one coordinating, one sort of killing cells, they're actually very closely related in this family tree. Although all these cells are created in the bone marrow, they don't necessarily all mature there. By that I mean reaching their final cell state. So some of these lymphoid progenitor cells will travel in the blood to the thymus, a kind of gland in the chest where they'll start their journey to becoming T cells. And that's where the T cells get their name, the T from the thymus gland where they mature.
制御性 T さん。
そうですか。今は T タイムではなく仕事中ですが。制御性 T
Right, so we learn about the regulatory T cells. Often don't hear much about them, sometimes called the Treg cells. And as it says here, they prevent killer T cells from spinning out of control. Don't forget, your immune system is a deadly thing. It is designed to kill things and would damage your own cells, what we call an autoimmune disease, if not well regulated. Part of this is down to the regulatory T cells, whereas most T cells activate the immune system, Treg cells inhibit the immune system. So the helper T cells is the coordinating, more of the maybe guy thinking with his brain, and the cytotoxic T cell, the guy thinking with his muscle, his job is to kill things. And the regulatory T cell, the one that inhibits the immune system, is more of a kind of relaxed, placid character. So, you know, they're totally well written to suit the role.
そう見えるよね。でも昔のあの二人はね。
Wait, so we're off down a trip down memory lane. We're going back to thymus school to learn about how the T cells develop. Welcome to hell, you useless runts. The flashbacks to med school. I'm kidding. And they've depicted the thymus here as this large, craggy mountain, not too dissimilar to how the thymus gland looks. The thymus sits just above your heart in an area we call your mediastinum. Worth pointing out at this point that the lymphoid progenitor cells, so the ones here from our diagram, have come, as we know, from the bone marrow, and they travel in the blood following chemical messages produced by the cells in the thymus. Once these progenitor cells reach the thymus, but before they become fully fledged T cells, we call them thymocytes, which is the name of this episode. So, you know, I'm guessing what we're going to learn all about. The thymus is where we're going to separate the useful from the useless. This is exactly right. It's pretty much summed up the job of the thymus. It has two jobs. Yes, you want T cells to be able to identify foreign antigens from germs. So an antigen is a piece of material, usually a protein, that your immune system uses to recognize things. But you must also crucially stop the T cells from attacking your own body's antigens. This development we call T cell education, and it's the primary role of the thymus. And it's pretty brutal. A thymocyte will have roughly a 5% chance of making it through this process without being killed. Quite a harsh education system. Cool, so exactly what you'd expect. We have the thymus epithelial cell giving the thymocytes a kind of run for their money, getting them into shape. The epithelium in general is the cells that surround our organs and tissues. It's kind of everywhere really, so on our skin, in our gut, inside of our blood vessels, and they're highly specialized depending on the job that they need to do. For example, the epithelial cells of the thymus here have special receptors on their surface in order to do this T cell education, so they'll positively select the thymocytes that they want. Cool. So we can see that there's a couple of other cells involved in the thymus as well. So we see the dendritic cell and the macrophage. They're also involved in this selection process. So nice little thing to include there. Wow, so only 2 to 3% will be standing at the end. It's even more brutal than I thought. Cool, so they're showing here the positive and negative selection, how the thymocytes become T cells and how they become which particular T cells. So let's get a little bit nerdy for a minute. So first up we have our thymus gland. So the outer part here we call the cortex, and the inner part here we call the medulla. That's kind of common terminology for lots of organs actually. Thymocytes arrive from the bloodstream in the outer layer in the cortex, and slowly as they mature work their way to the medulla, the center, where they eventually become fully fledged T cells and released back into the bloodstream to do their job. So here we have 3 thymocytes that have arrived into the cortex and they want to mature into T cells. You can see at first they're quite shy and they don't really have any receptors on their surface, but that's soon to change and they come out of their shell a little bit. Pretty much what we see here in Cells at Work. So the main receptor they're going to display is the T cell receptor. And we're going to talk about some other co-receptors as well. They're things that help that receptor bind to its target. So they all display a unique T cell receptor. And this is randomly generated, and its whole point is to recognize antigens presented to it. So each has a unique T cell receptor, but they also have on their surface common co-receptors called CD8 and CD4. So the only thing that makes them different at this point is their T cell receptor, and this is represented in Cells at Work by our thymocytes early on, looking very similar with their uniforms. It is the change in these co-receptors that determine what kind of T cell they'll become. So if they solely displayed the CD8 co-receptor, they'd be a cytotoxic T cell. And if they solely displayed the CD4 co-receptor, they'd be a T helper cell. So who figures out what co-receptors they're going to display and therefore what T cell they're going to become? Well, we know already it's the drill sergeant, it's the thymus epithelial cells. So here is our epithelial cell of the thymus gland. So these drill sergeant epithelial cells display 2 types of similar molecules on their surface, MHC1 and MHC2. We won't worry about the complex names of these, but these molecules are what communicate with the T cell receptor. So it's super important that the T cell receptor is able to bind to them properly. And depending on what MHC receptor the T cell binds to will determine what type of T cell we will get. MHC1 receptors are not just found on the thymus epithelial cells, they are pretty much found on all the cells in the body apart from our friend the red blood cell, and are used to tell immune cells what this cell is up to. So most of the time the cell would just have its own proteins on its surface, basically indicating that it's a healthy cell. But if the cell was infected with a virus, it would contain viral proteins on these receptors, saying it had become damaged and therefore we'd want to kill the cell. Therefore, MHC1 molecules you want to be easily read by cytotoxic T cells because they're the ones able to kill damaged cells. This is why if the thymocyte T cell receptor binds strongly to the MHC1 molecule, we will get signals to upregulate more of the CD8 co-receptors and less of the CD4, meaning that we're likely to convert this into a cytotoxic T cell. So at this point, we're not worried about the actual antigen protein being presented to the T cell. We just want to make sure that this T cell receptor works really well with this MHC molecule. Conversely, the MH2 molecules are only really found on immune cells such as macrophages, neutrophils, and B cells, the type of cells that helper T cells activate. Therefore, if the T cell receptor binds really strongly with the MH2 molecule, therefore upregulate the CD4 co-receptor, meaning this will begin to turn into a T helper cell. So we have this thymocyte has bind to the MHC1, so it's likely to become a cytotoxic T cell. This thymocyte has bind to the MHC2, so it's likely to become a T helper cell. But this one here doesn't bind to any of these MHC proteins. Well, it becomes destroyed. Because it cannot identify the crucial receptors it's supposed to communicate with. This whole process is called positive selection, as the strongest ones are kept. So now we have all our T cells ready for action, right? Well, not quite. They then move from the cortex to the medulla, the central part of the thymus, where negative selection happens. These single positive thymocytes By that I mean they either at this point have CD4 or CD8 exclusively, are then exposed to your own body's antigens to make sure they don't react with them. And we see this beautifully illustrated in Cells at Work. So we see here the macrophage holding up an effigy of one of the own cells and seeing if the thymocyte reacts to it. If it does, it's killed. You don't really see that, you just see a little trapdoor, but it's disposed of. And this is pretty much exactly what happens. So macrophages here found in the medulla of the thymus, and they would have picked up lots of antigens from your own body on its travels, and it lays them all out for the T cells, the T cell receptor to detect. And if one of these developing thymocytes strongly reacts to that protein, then we know it's going to react to your own body, so it is sadly killed. It goes down the chute. But if it only weakly binds to it, then it's considered a pass. It's not strong enough to damage your own tissue. So this would be the T cell that ends up getting produced. So this whole process in the medulla is called negative selection, because the ones that react the weakest are the ones we want, and the ones that react the strongest are the ones we kill. So wow, we managed to get through it. I hope that kind of made sense a little bit. So in our little example, this helper T cell is the only one that's made it through into the bloodstream to do its job. So we find out just how cytotoxic T cells kill the cells using the ultimate internal secret art antigen bludgeoning pulverizing fist. And I thought it was the fast ligand, but you learn something every day. Yeah, embarrassing photos of your immature times. Who would ever share that type of thing? So there you have it, guys. My look at episode 9 thymocytes of Cells at Work. I remember last week saying there wasn't much immunology last week. They certainly made up for it this week, but it was a great way to visualize the role of the thymus, you know, to protect the development of these cells so they don't get released into the body and cause you loads of damage. I thought that was done absolutely brilliantly. And I know there's always so much detail in these, so if there's anything I've missed, let me know, because I tend to sort of then try and feed that into future videos and make sure everyone's aware of, you know, quite how clever this is. So it just leaves me to say thank you so much for all your support. I hope you've enjoyed this video. If you have, give it a like and a comment, or maybe subscribe to the channel if you haven't already. And to everyone that has, thank you so much. We've hit 350,000 subscribers. Mind is blown. So until next time, guys, I'll see you soon.